Thursday we'll be practicing y'all's chalk talks. How are those coming along?
Good.
Okay, well, I will go through my chalk talk for y'all today. And this is like a good opportunity to ask any questions you have. Bear with me, I have not done this in like 2 years. So, okay. Hi everyone, I'm Marina. I'm doing my dissertation and research in the Examples Lab in the Department of Cell and Developmental Biology, where I am investigating the role of mitochondria in neural development. So mitochondria are well known and characterized for their involvement in metabolism, energy production, and notably apoptosis. And in the early 1990s, a group of scientists in Craig's lab found that this novel, at the time, protein called MCL1 that they isolated from myeloid cell leukemia. And it is a protein that's abundant in cancer cells. And that is for its involvement in inhibiting cell death, specifically apoptosis at the outer mitochondrial membrane. However, another group in the early 2000s, when they knocked down MCL1 gene in blastocysts, they found that these embryos never attached. So there was an absence of, it was essentially fatal, it was fatal at the embryonic stage. And when they isolated cells from the inner cell mass, these are the stem cells that make up the embryo, they did not see an elevation in cell death, suggesting that MCL1 plays another crucial role in development that is not due to its canonical anti-apoptotic role at the mitochondria. These ICM cells in culture we call human embryonic stem cells, or H9s, which I'm using to derive neural progenitor cells in culture. Now, these neural progenitor cells are positive in PAC6 and SOX2 and nestin, which are all identity markers of NPCs. However, when I knock down MCL1 or inhibit it in these NPCs, I see a downregulation of PAC6, nestin, and SOX2. Without a notable increase in cell death, suggesting that MCL1 in neurodevelopment is playing some role in cell fate decisions at the mitochondria. Now, what can that be? Recently, in 2018, another group found that there is a shortened form of MCL1 found at the inner mitochondrial membrane. And work from our lab has shown that MCL1 may be interacting with OPA1, which is important for the formation of this cristae. And something notable about the cristae is you have ATP production here, and you also have the Nico's complex protein, which is involved for maintaining this shape. So OPA1 stabilizes this tube section of the cristae, and Nico's, when it's knocked out, you no longer have cristae invaginations. So perhaps MCL1 at the IMM is playing a role in maintaining cristae morphology and energy production in NPCs, hence why we see the loss of identity. And so for my aims, I'm looking to identify the function of MCL1 in HNPCs, and I'm proposing that it is involved in cristae morphology and ATP production. And for my second game, I'm interested in characterizing the mechanisms by which MCL1 does this. So the mechanisms of MCL1 in HNPCs. And I'm proposing that this is due to a potential interaction between MCL1 and OPA1 and or MCL1 and the mucose complex for the maintenance of the crystal. I don't remember my closing impact statement, so we will end it there. But yes, I did have a closing impact statement. And I did make everything pretty big. I fit the whole board because then I was asked about the trophoblast layer, and I was asked to specifically draw out the different mucose proteins, so things like MIG-60, MIG-19, etc. So yeah. Any questions?
Nothing.
We want to jump into
I think so. Yep.
Then we can do development and then go into non-formal.
Yep. So I'm gonna go ahead and erase this. Something I did for my chalk talk is I drew it out like this, and I went and practiced on the board, and I would write down what point I'm going to make next. So I like literally numbered, okay, this is how I'm gonna start, and I wrote that down. So like really get up on board and practice. I encourage the use of like one or two different markers, like for different colors to point out really important things. So everything with MCL1, I had made it red, for example, but you know, limited to one color today. Anyways, just some suggestions as you prepare for next week's talk talks. So I'm gonna start with neural tube formation and the 3 germ layers. Can anyone tell me what the 3 germ layers are? I'll just have them as lines. Okay, can someone tell me the order?
Ecto, meso, endo.
Exactly. And how do you remember that?
Ecto meaning like your outside, meso middle, endo meaning
Exactly. Now, which one of these is responsible for neurodevelopment?
ectoderm.
And so we will not need this one, so I'll get rid of endoderm. Yes, so ectoderm is really important. You'll have here your neuroectoderm. And this kind of starts to fold in half. And what essentially happens,
But does anyone know what the area outside of the neuroectoderm becomes?
like here,
Yeah. And then what signaling dictates that?
Is it, I don't know, is it the tube or the plate? Or
No,
the
what-
crest?
so
One
the
of these ones.
neuroectoderm will make your entire nervous system.
Okay.
What about the ectoderm layer? What's outside?
Skin?
Do you know what signaling dictates that? There's a morphogenic signaling that occurs during development.
Yep.
Specifically BMP4 inhibition.
Is it bone morphogenic protein?
Yep.
Bone, that's the signaling pathway.
So this neural here will begin to fold at the medial hinge point. So let's call this is the middle, and this is the medial hinge point. And what you start to get is this dent. And this is going to now be your neural fold. And you have here your notochord, which will become very important later on. And just know, don't forget, just keep in mind, because I don't want to keep having to erase it, that the mesoderm is right under it, okay? So then from neural fold,
Go.
this is neural plate, you'll have neural root, now these two will essentially fold in on each other. And these would be does anyone remember what these are expressing? Or another name for them? So if this is your medial hinge point, what would these two be? Dorsolateral hinge points. Eventually, when this fully closes, it will separate from your ectoderm. And this is now your neural tube. And your notochord spreads out and become what? So as a hint, the neural tube is responsible for making your central nervous system.
Does anyone know what resident neural cell is not derived from the ectoderm? We talked about it last
Is it glia?
week. Only one specific type of glia. Do you know where they come from in development? Anyone else know? They don't come from the ectoderm. But they end up residing in the brain.
What do they do? What do microglia do in the brain? I'm gonna help you. We reviewed this last week. What are the microglia doing in the brain? What are they responding to?
There's finding different cytokines, because then they have like uptake for phagocytosis, they're the resident macrophages of the brain.
And so,
Resident macrophages or resident immune cells?
Immune cells.
exactly, so they are from,
They have to be mesenchymal.
So they're derived from the yolk sac. Yeah, so pretty different, but they penetrate the brain through the vascular system, which derives from what layer? Yeah, there are only 3 layers to choose from, so someone start saying something.
Yeah.
Mesoderm?
Endoderm.
No.
Mesoderm?
mesoderm.
Yes.
Mesoderm.
Okay.
So the reason why I say the mesoderm is really important is because it actually has signaling to direct the neural crest cells to where they are to go. And this is also important for another type of nervous system we don't talk often about. Michelle Southard Smith
The
studies it.
enteric.
Yes, your enteric nervous system. Okay, so also as a fun fact for neural crest cells, they also make your melanocytes. So it's not just your nerve cells. So any pigmentation, neural crest cells. All right. So now, does anyone remember what the Hox genes are? Jump forward a bit.
What do the Hox genes do? Let's start with that. Like, why do we know them? Why are they important?
So we can take a
I only
closer look.
remember like flies having like eyes on different parts of its body and stuff like that. I can't remember anything else
About
about Hox genes,
Yeah.
like the location,
Yeah, you're on the
like
right track.
generation of cells or like organs.
It's really important for organizing your vertebrae.
Okay.
So if we think about the central nervous system, what are two main organs we think of? Brain
Brain.
and spinal cord. The spinal cord, this is my vertebrae, everyone. Yeah, and so essentially with the neural tube, you'll have what's known as neural tube patterning, where you have expression of different Hox genes, and they're just numbered. And those are responsible for the organization and the cell types that you will have in the vertebrae. And now focusing on the brain, we have neurogenesis. So can anyone tell me the different layers in neurogenesis? Rebecca Erie gave that lecture my year of fundamentals. I don't know if she's still the one doing it. She isn't? No.
The only development is Howard Smith.
I see. Okay. So who remembers something like this? Let me get y'all started. This is your ventricular zone. There will be cortical layers. I think I do. And which order are they labeled? And versus which order do they develop?
I would say, just guessing off the slide picture, it goes bottom to top 1%.
One is here or here? One is at the top or the bottom?
Oh, not one at the bottom. But what I'm thinking, but I don't think that's.
Okay, so this is interesting because they're labeled as layers 1 through layer 6. Powever, they don't grow from an outside in, they grow from inside out. The reason that is because early on during histology, this is how the different layers were labeled from the top to bottom. However, that's not how they develop. They develop from inside out. So those are your cortical layers. Can someone help me finish out these zones? There are two, and one of them is really important for distinguishing kind of sort of a developmental difference between mice and humans. And I say sort of because that's been debunked, but you know, faculty members, yeah.
there like a periventricular? Or am I thinking of something completely different?
They all have the word. There is one that has a ventricular. So this is your ventricular zone, and then outer and intermediate. So you have your outer subventricular zone. That's not in the book, but I remember it. Your outer zone, outer ventricular zone, and your intermediate zone. So ventricular, outer subventricular is between the ventricular and outer ventricular zone, and then your intermediate is between your outer ventricular zone and your peripheral layers.